Kratom. We will hear first from Dr. Marilyn Huestis, who is a professor, retired as a tenured senior investigator and chief of chemistry and drug metabolism section at the National Institutes on Drug Abuse. The National Institutes of Health in 2016 retired there after 23 years of conducting controlled drug administration studies. Currently is with Thomas Jefferson University, Pinney Associates, Huestis, and Smith Toxology. Each of these experts will have a more robust and deserved credits to their biographies posted along with the recording of this. But first, we'll talk to Marilyn Huestis, who is joining us from a ship out in the middle of the ocean. But she, I think it's about 3 in the morning for her, but we welcome her to participate in this event. Thank you, Marilyn. We'll turn it to you.
Well, good afternoon, everyone. And we're very excited, all of us, to be a part of this. We're going to be talking about both rat and human studies on the safety, abuse, liability, pharmacokinetics, and toxicology of kratom. So kratom is a Southeastern Asian evergreen tree that's related to the coffee family, and the leaves of this herbal medicine have been used for hundreds of years in traditional medicine. Kratom contains many alkaloids, and mitragynine is the most prominent one, representing about 1 to 2% of the plant weight. It is a partial mu opioid receptor antagonist, but it does not activate the important beta-arrestin 2 pathway that is involved with respiratory depression and constipation. And it has a wealth of other receptor activity that makes it distinct from opioids. It has this unique pharmacology that suggests it could be a safer analgesic and also a potential treatment for opioid use disorder. There is tremendous new research data, a lot of which you'll hear about today, that is sponsored by the National Institute on Drug Abuse and many university centers in Southeast Asia. We're going to be talking, all 4 of us, about the reasons for abuse, the abuse potential, withdrawal, potential treatment as an analgesic and substance use disorder, and a lot more about the pharmacodynamics and pharmacokinetics. I'm going to first talk about a study that Dr. Henningfield and I conducted, where we compared the respiratory depressant effects of mitragynine and used oxycodone as the positive control, as suggested by the Food and Drug Administration for studying this type of effect. We received support for this study from the Center for Plant Science and Health, which is a foundation of the American Kratom Association. But we want you to know that neither of these organizations had any input on the design, analysis, or publication of these results. Rats received up to 400 milligram per kilogram of oral mitragynine, and those effects were compared to the therapeutic dose of oxycodone, 6.75 milligrams per kilogram, and 2 higher doses. We then measured blood gases in the rats after dosing, and also their pharmacokinetics. And what we found was a significant reduction in the oxygen saturation in the rats, where compared to mitragynine up to 400 milligrams per kilogram, we showed no significant effects. There were also 2 deaths, 1 each in the high oxycodone doses and none in the mitragynine. We also showed a significant increase in the partial pressure of carbon dioxide in the high oxycodone doses, and again, no significant effect with mitragynine doses. And this was the same for all of the different mitragynine, all the other blood gas measures. Also, this is not novel results. We know that the same lack of respiratory depression has been shown in 4 other animal species. Now, I'm excited to share some results on a very large, complex study that was conducted in 198 healthy adults. It was intensive and involved 30 clinical site visits per subject over 47 days. We looked at multiple aspects of mitragynine safety issues, and also looked at a number of parameters assessing the abuse liability, including the clinical opiate withdrawal scale, or COWS, and the subjective opiate withdrawal scales. And I'm going to share with you some of the very first pharmacokinetic data as well. Now, these COWS and SOWS instruments look at abuse liability during the potential withdrawal period. And so we had 15 multiple doses, once each day. And after the end of those 15 doses, we found no sign whatsoever of opioid withdrawal with these instruments. Now, I only have the first two cohorts available today. The study recently finished, and we're waiting for the results from the two higher doses. Here are the single dose pharmacokinetics for metragenine and its more potent metabolite, 7-hydroxymetragenine. And you can see a nice dose orderly increase with the doses up to 53.2 milligrams. And these are after single doses the first 24 hours, but we monitored for 7 days after dosing. And then after a repeated dose, daily doses of metragenine, you can see here the C maxes for the 4 different doses, the maximum concentrations, and when they occurred for the parent drug metragenine and the 7-hydroxymetragenine. There were no adverse events in the healthy volunteers that received these doses up to 53 milligrams per day for 15 days. Now, those data are important because they shed light on the concern about potential metragenine overdoses. And as I said, we had no adverse effects at these doses. So what about the potential for kratom to produce overdoses and deaths? Well, none of the 6 major U.S. surveillance systems have identified kratom as a public health threat. We have just heard from Mac Hadow that the Department of Health and Human Services withdrew its official request for scheduling. And as you know, the World Health Organization also did a recent, very thorough review of metragenine and 7-hydroxymetragenine, and they advised the commission that there was insufficient evidence to schedule kratom at the international level currently. So unfortunately, in most, but not all cases where metragenine is included in the list of cause of death, there are numerous other respiratory depressant drugs that are found in concentrations that could contribute or result in death. Unfortunately, there is limited toxicology performed in many cases, and this is because when they find metragenine at the scene where it may be used to treat opioid use disorder, they may limit the toxicology to measuring metragenine and not look for other substances that could be contributing. One of these major areas is with NPS, or novel psychoactive substances, which are frequently found in these reported deaths. And this requires that the sample be sent out to a reference lab, usually, to test for these. And the cost involved prohibits poorly funded forensic labs in many cases from doing this. Another major issue is adulteration of the metragenine itself. So there are cases published that 7-hydroxymetragenine, the metabolite, potent fentanyl, O-desmethyltramadol, or other contaminants could produce these deaths. And so adulteration is a major issue. So we suggest that it's much preferable to regulate kratom quality and prohibit adulteration than to ban kratom and risk the increased potential for opioid overdose deaths. I have some references, and I'm happy to turn it over to Dr. Kirsten Smith. Thank you.